acid-base balance of blood. This acid-base balance is essential for the normal life activities of the body. Normal pH of the plasma is 7.4 and it should be maintained within 7.38 to 7.42. If it is too less, it will lead to acidosis. If it is too much, it will lead to alkalosis, and both are extremely dangerous. How many methods are there in the blood to maintain pH? There is a first line of defense, blood buffers are there. There is a second line of defense, respiratory regulation is there. There is a third line of defense, there is the renal regulation. We can see here the buffer systems of the body. Body buffers. You see, buffer is the first line of defense. So, which are the buffer systems of the body? In our body, in the ECF, there is the buffer system, carbonate buffer and phosphate buffer. Bicarbonate buffer and phosphate buffers both are there in the ECF, extracellular fluid. You know that blood is actually extracellular. And in the intracellular fluid, you have phosphate buffer and also protein buffer. While inside the erythrocyte, you have hemoglobin and also phosphate. So you see, in all the cases, phosphate is there. And first of all, let us come to the case of bicarbonate buffer system. This is a very important buffer system. Sodium bicarbonate and carbonic acid buffer system is there. It accounts for almost 65% of the buffering capacity of plasma and 40% of the buffering action of the whole body. You know why? Because this carbonate can be generated from carbon dioxide and its concentration can be regulated in the body, can be maintained in the body by way of respiration. Therefore, concentration of this buffer and this buffering action can be very well maintained. The base component, the bicarbonate, is regulated by the kidney by means of metabolism, while the acidic component, carbonic acid, is under respiratory regulation. So you see the different pH buffering systems are interconnected and they are interdependent upon each other. You can see here the normal bicarbonate buffer etc. are given here, and presence of bicarbonate in relatively high concentration is very much useful for the system, and the components are under physiological control. Carbon dioxide is regulated by lungs and bicarbonate is regulated by kidneys. Excretion of the hydrogen ions, the protons, in the proximal tubules of the kidney. You see, alkali is required to the plasma, bicarbonate. From the PCT, sodium ions are reabsorbed into the plasma, while these sodium ions into the PCT, proximal converted tubules of the nephron, are coming from the tubular lumen. You might have heard of the stages of urine formation. First of all, there is ultrafiltration, then there is tubular reabsorption, and finally there is active secretion. These are not mentioned in this slides, but in your lower classes you might have heard of it. So from the tubular lumen, after the ultrafiltration, sodium ions are absorbed first into the PCT cell and then into the plasma. While in the PCT, carbon and the PCT cell, carbon dioxide reacts with the water forming carbonic acid. What is Ca? Ca is carbonic anhydrase, the enzyme required for this. This carbonic acid is dissociating into HCO3 minus and H plus. The H plus is secreted into the lumen. H plus, the protons are secreted into the lumen. That's why actually urine is slightly acidic in nature. And the basic component, alkaline component, bicarbonate, is actually transported into the plasma. This is regulated because this bicarbonate is forming the buffer component of the blood. While what is this one? Are they the same component. They are not the same component actually. Reabsorption of bicarbonate from the tubular fluid. You see, as we mentioned earlier, from the tubular lumen, sodium ions are taken into the tubular cell and then into the plasma. And how this sodium is reaching there when from the sodium bicarbonate. How the tubular lumen contains sodium bicarbonate by means of ultrafiltration in the nephrons. So the sodium bicarbonate from the blood is filtered into the lumen, and there it is decomposing or ionizing into sodium ions and bicarbonate ions. Sodium ions immediately enter into the tubular cell and then into the plasma. And what about the bicarbonate? The bicarbonate is combining with the protons which are coming from the tubular cell. In the previous slide also we found that by the ionization of carbonic acid, we are having protons, and the protons are secreted into the lumen. This bicarbonate again combined with this protons forming carbonic acid. This carbonic acid is acted upon by carbonic anhydrase to produce carbon dioxide and water, and depending upon the necessity of the body, sufficient carbon dioxide is entering into the tubular cell. Remember, depending upon the necessity. Okay? Yes. And phosphate buffer system. It is mainly an intracellular buffer, even though it is present in the other components as well. And it is found to be very much effective at a wide range of pH because it has more than one ionizable group. You might have heard of phosphoric acid, H3PO4, then HPO4 minus, H2PO4 minus and HPO4 2 minus. So it has more than one ionizable group, and the pKa values are different for both of them. And in the body, disodium hydrogen phosphate and sodium dihydrogen phosphate, these are interchangeable in each other, and this is an effective buffer system because its pK value is nearest to the physiological pH. And a protein buffer. Buffering capacity of protein depends upon the pK value of the ionizable groups. The most effective group is the histidine imidazole group with a pK value of 6.1. In the case of albumin also, in a slide you might have seen that many histidine groups are there in albumin. It is constituting, it is contributing a lot towards pH maintenance. In the body, 52% of the buffer activity is in the tissues and 6% in the RBCs, and 43% in the extracellular buffer. So all components of the body are contributing towards maintenance of the pH. The same thing in a diagrammatic representation. What about the respiratory relation of the pH? This is achieved by changing the partial pressure of carbon dioxide, PCO2. Remember, carbon dioxide's concentration can be regulated in the blood by way of the respiratory rhythm center of the brain. Carbon dioxide concentration is very much sensitive. Body is sensitive to the concentration of carbon dioxide, and its concentration can be regulated in the body. So you just remember, carbon dioxide is not merely a waste material. Don't believe that carbon dioxide is only a waste material. It is useful for the body. It is used as a buffer. It is used for the preparation of buffer. And the extra carbon dioxide is released out of the body by means of expiration. That's all. When there is a fall in pH of the plasma, that is acidosis, respiratory rate is stimulated, more carbon dioxide is given out, so carbonic acid level can be reduced. So you see how much simple it is, but it is not that much simple actually. And action of hemoglobin. Hemoglobin is there inside the RBC. It is used for the transport of carbon dioxide. That way it is important in the pH of the blood. What is the role of carbonic anhydrase? It is combining carbon dioxide with water forming carbonic acid, and then the carbonic acid will dissociate into bicarbonate ions and the protons will combine with the hemoglobin and it will produce this HHB. HHB means the hemoglobin is carrying protons, so hemoglobin is a buffer. It will combine with the protons. So this is the first one, and the reverse is occurring in the lungs. It is here. This is the one, and this is here. In the lungs, what happens? HHB is coming from the tissues to the lungs. In the tissues, hemoglobin is combining with the protons, forming HHB. That HHB is coming to the lungs in the blood circulation. In the lungs, this is reacting with oxygen, producing oxyhemoglobin, and the protons are given out. And the protons combine with the carbonic acid, bicarbonate ions, forming carbonic acid. Carbonic acid dissociates into carbon dioxide and water, and both are given out through the exhaled air. You see, these things are coming out through the exhaled air. You know what is this? This is your eye. So you are expiring, giving out carbon dioxide. So this is a way of transporting protons by means of hemoglobin. Three, renal regulation of pH. By now you might have understood that protons are regulated by the kidneys. Proton concentration is regulated by the kidney. How? Excretion of protons, reabsorption of bicarbonate ions, excretion of titratable acid, and excretion of ammonium ions. So kidneys are playing a very important role in the regulation of pH of the body. So you can just imagine what happens in case of a serious kidney damage. In case of a serious kidney damage, all these functions are affected. First point is excretion of protons in the proximal convoluted tubules of the nephron. Carbon dioxide combines with the water, forming carbonic acid. We have seen all those things, and we have seen this in a diagram as well. Reabsorption of bicarbonate, this also we found in a diagram previously. Bicarbonate is reabsorbed. Excretion of titratable acid. In the DCT, net acid excretion occurs. Protons are secreted by distal tubules and collecting ducts by H plus ATPase located in the apical cell membrane. The H plus are generated in the tubular cell. You have seen earlier by the decomposition of carbonic acid by carbonic anhydrase, etc. The term titratable acidity of urine means it is the total number of milliliters of N/10 sodium hydroxide required to titrate 1 liter of urine to pH 7.4. How many milliliters of N/10? That means 0.1 normal NaOH. How many milliliters of 0.1 normal NaOH is required to titrate 1 liter of urine to a pH of 7.4? That is what is called titratable acid present in the blood. Why 7.4? Because we believe that 7.4 is the optimum pH. Okay, the major titratable acid in the urine is sodium acid phosphate. Okay, and it is formed from disodium hydrogen phosphate, basic phosphate, etc. Acid and basic phosphate pair is considered as the urinary buffer. The maximum limit of acidification is pH of 4.5, inhibited by carbonic anhydrase. Yes, phosphate mechanism. We found actually this earlier. Sodium dihydrogen phosphate present in the lumen is broken down into sodium ions and monosodium hydrogen phosphate. Sodium is entering into the tubular cell and then into the plasma, and this way protons are taken from the tubular cell in the tubular lumen. They are combining with the monosodium hydrogen phosphate and again forming sodium dihydrogen phosphate, and they are excreted. So by this way the excess protons are removed. In the form of sodium dihydrogen phosphate. And excretion of ammonium ions mainly in the distal convoluted tubules DCT. This should help you excrete protons and reabsorb bicarbonate ions. And the mechanism is explained here. There is the glutaminase present in the tubular cells. It can hydrolyze glutamine to ammonia and glutamic acid. The ammonia diffuses into the luminal fluid, combines with the protons there. You know that protons are excreted. And then they are forming ammonium ions. Remember, ammonia combines with the protons to form ammonium ions. The glutaminase activity is increased in acidosis. In acidosis means more protons are there. Therefore, glutaminase is excreting more ammonia. This ammonia combined with the protons forming ammonium ions, and these ammonium ions are excreted. That way, more protons can be removed from the plasma. Okay, the same thing is actually mentioned here in this diagram. This is the summary of the buffering action. Whatever we found so far can be seen here. Just go through it. And that is the end of this chapter.